If this is too distracting, let me know. I'm willing. Oh, and one more announcement. Almost forgot. Today at noon at CST 103, the first of the biology senior seminars this semester will be presented by Lily Hartman, Madeline Chomentowski, Sebastian Slavinskas, and Brady Bubb on what's going on down there, community ecology of caves. So many of you are seniors, some of you will be doing this in the future. Here's the first senior seminar of the academic year. Pizza, as always, please attend if you can. Cave ecology, new today. All right, so today what we're going to do, no, I have to do this. Today what we're going to do is go over, review the transport mechanisms for reabsorption of sodium and glucose, and then do some clickers, I think, and then move on to bicarbonate, and then potassium. So remember that 2/3 of the sodium that is reabsorbed in the proximal tubule. The proximal tubule is leaky, so the reabsorption of sodium is accompanied by substantial reabsorption of water. We're going to be talking much more about water on Tuesday. But a lot of the water that you reabsorb is in the proximal tubule, simply following the active reabsorption of sodium. Then some of the sodium is reabsorbed in the ascending limb of the loop of Henle, about 1/4 of it, and then the rest is reabsorbed in the distal tubule and the collecting tubule. Remember that we filter sodium, but we need to reabsorb it or else we die pretty quick. Same with water, we would dehydrate almost in minutes if we didn't reabsorb water from the filtrate. So this was the challenging slide from last time. Did everybody get a handout today? They were passed around. Okay. So let's go through this one more time, maybe with a different pattern. Let's look at the commonalities and differences in the different segments of the tubule, different tubular segments. So here is the proximal tubule, here is the ascending loop of Henle, here is the distal tubule, and here is the collecting tubule. So 4 of the segments of the nephron. So one of the commonalities is that they all have a sodium-potassium ATPase in the basolateral membrane. And that sodium-potassium ATPase is working hard, pumping sodium out and potassium in. So that's an important player in sodium reabsorption in all segments. Basolateral expression of the sodium-potassium ATPase. In the proximal tubule, in addition on the basolateral membrane, we have a sodium bicarbonate co-transporter that also participates in sodium reabsorption of the basolateral membrane. Remember, reabsorption is moving sodium from the luminal fluid, the urinary fluid, into the interstitial space or the peritubular space, where it will then be picked up by peritubular capillaries and recycled back to the body. So we're moving sodium from yellow to blue in all of these images. We pump sodium with the sodium-potassium ATPase in the basolateral membrane, and then we have the secondary active transporter also for movement of sodium in the basolateral membrane in the proximal tubule, but not in any of the other segments. We're going to see this again when we talk about bicarbonate in a few minutes. So how do we get sodium across the apical membrane? Well, in the proximal tubule, we have a sodium-glucose co-transporter. and that's going to be important for glucose reabsorption as well. In the thick ascending limb of the loop of Henle, just ascending limb of the loop of Henle, we have a sodium potassium 2 chloride co-transporter that moves sodium in. In the distal tubule, we have a sodium chloride co-transporter that moves sodium in. And in the collecting tubule, we have a sodium channel that moves sodium in. So those are some, not all, but many of the transporters in the apical membrane. Sodium glucose, sodium potassium 2 chloride, sodium chloride, and sodium channel. In addition, there is a sodium proton exchanger here in the apical membrane of the proximal tubule. The water that is reabsorbed in the proximal tubule is paracellular through the leaky junctions. Okay? So those are things to know. For glucose, it's simpler-ish. Yeah, it's simpler. Sodium, potassium, ATPase on the basolateral membrane in the proximal tubule as everywhere else. We only have to look at the proximal tubule because it's only in the proximal tubule that we express a sodium glucose cotransporter. Whether it's sodium glucose cotransporter 2 or sodium glucose cotransporter 1 is a detail that is not necessary to memorize. But sodium and glucose co-transport into the cytoplasm, driven by the sodium gradient created by the sodium potassium ATPase, and then glucose moves passively by a uniporter, and whether it's GLUT2 or GLUT1 doesn't matter for this class. But you do need to know that there is a uniporter, a passive transporter expressed on the basolateral membrane of the proximal tubule, which carries glucose from cytoplasm into the interstitial space where it is then picked up by the peritubular catheters. Okay, let's do it quickly. You want me to stay in character, or should I get out of here?
What do you want to do?
Whatever you want, bro.
Oh, I do.
Yeah, I'm Professor X, so I can do whatever.
I love it.
I didn't think to be asking you if we could be doing a think, pair, share about what your superpowers are. Anyway, let's do a clicker. Okay, I think I got them all right. I think they're all true. I saw a lot of you had them all, had the whole sheet up. Are there any questions about those of you who didn't have the all sheet up, who missed one of these questions? Happy to explain it.
Can you pick up
C. So at the end, so C is a little tricky. At the end of the proximal tubule, in a normal person, the glucose concentration in the tubular fluid is 0. Because if it were not 0, it would be in the urine. And for a typical person, the concentration of glucose in the urine is 0. And since the proximal tubule is the only place where glucose is reabsorbed, the concentration of glucose at the end of the proximal tubule is 0. So, so you could say, well, okay, you might not have circled C if you thought it was 0 in both the tubular fluid and in the peritubular space. That would be a legitimate explanation for why you would say C is not true. It's not less than, they're both the same, and they're both 0. But 0 is hard to obtain. 0 here is hard to obtain. 0 here is easier to obtain because of this high affinity sodium glucose pump for glucose. So here the concentration of glucose is probably not quite 0. And here the concentration of glucose is 0, typically. And that's what that was getting at. And since something is always greater than nothing, 0, that's why that's a true statement. Does that work for most of you? Yeah. 0 is lower than not 0. Okay, let's see what comes up next. So the point is that if the concentration of glucose at the end of the proximal tubule is not 0, it will get cleared, it will get excreted. That's what happens in diabetes when the concentration of glucose in the plasma is high, because that's the condition of diabetes, is the concentration of glucose in the serum is high. If it's high enough, that is to say greater than 250 milligrams per deciliter, it spills over it gets past the proximal tubule, and then it's present in urine. The excretion of glucose is not really what makes diabetes a disease, a disease that causes a lot of problems, including blindness and amputation of limbs and short lifespan. The high glucose in the serum is the reason why diabetes is a debilitating disease, and that high glucose causes a lot of things to happen. Vasoconstriction, poor circulation in the limbs, poor circulation in the eyes, leading to blindness. It's the high serum glucose that causes the disease of diabetes, not the simple fact that you're urinating it away. Okay, let's do another one. I think B and D are the correct answers here. So let's see. I think most of you got those. Some of you added C. Now, excessive reabsorption of glucose, what causes the clearance of glucose to be greater than 0 in diabetes is insufficient reabsorption of glucose, not excessive reabsorption of glucose. C would be true if it said insufficient reabsorption of glucose causes the clearance of glucose to be greater than 0. Remember, clearance is what happens when something is in the urine. And glucose is not cleared normally. So in a normal person, the clearance of sodium is less than the clearance of glucose. No, that can't be right. Glucose, in a normal person, the clearance of glucose is 0, and we can never have a negative clearance. The reabsorption of glucose by the proximal tubule depends on the presence of sodium. Yeah, we have the sodium-glucose co-transporter. I did see. Glucose is freely filtered. So this is what happens in diabetes. A high plasma concentration of glucose causes a high concentration of glucose to enter the proximal tubule. That's diabetes. Questions about these? All right, I'm not going to draw on the board. I'm going to try to stay in character. Let's switch gears and talk about bicarbonate. We talked about bicarbonate in the respiratory section. We sort of black boxed the idea that the kidneys can excrete bicarbonate. Remember the Davenport diagram where renal compensation for hyper or hypo ventilation were there. We'll see that again in a minute. Now we're going to open up that black box about how the kidney reabsorbs or excretes bicarbonate. Okay, so here's a figure from your textbook. Let's get oriented. Here is the urinary fluid. Here are the epithelial cells, just 2 of them being shown here. Here are the junctions. So this is the apical membrane, which should be zigzaggy, to illustrate that this is a sort of a brush border, it's called. And this is the basolateral membrane. So I blanked out some things that we want to talk about. Let's begin here where it says begin. There is bicarbonate in the filtrate that enters the proximal tubule. We can say, let's just say we're in the proximal tubule, it's simpler, but in fact all of this is valid throughout the kidney. So let's just talk about it as if it were proximal tubule. Bicarbonate is small enough to be filtered by the glomerulus, so it enters the filtrate, so it enters the proximal tubule. So what word goes, and you know from the Davenport diagram that the typical amount of bicarbonate in plasma is 26 millimolar, so 26 millimolar is filtered. And so what is present in the filtrate is 26 millimolar, or so. What goes in the box? What is the mechanism by which bicarbonate is in the filtrate? It's a simple question. Filtration. This is filtered bicarbonate. That's how it got there. That's where we're going to begin. Okay, what is the name of the enzyme that converts carbonic acid into water and carbon dioxide? Carbonic anhydrase. Now, what is this carbonic anhydrase doing out here in the fluid? The answer to that is there is a form of carbonic anhydrase called carbonic anhydrase 4, Roman numeral 4, CAIB. You don't have to know the different types of carbonic anhydrase, just like you don't have to know the different types of sodium glucose cotransporter or glucose uniporters. But you do need to know that there is a carbonic anhydrase that is expressed on the apical membrane, and it's a membrane-bound form of carbonic anhydrase, and its enzymatic, its enzymatic site is out extracellular. So it does its thing by being tethered to the apical membrane, but it's enzymatically acting on carbonic acid here. So what goes in here is carbonic anhydrase that is expressed on the apical membrane and has its active site facing the tubular fluid. There is also carbonic anhydrase in the cytoplasm. It's carbonic anhydrase, Roman numeral 2. No, carbonic anhydrase 2 is a cytoplasmic carbonic anhydrase in the cytosol of all of these epithelial cells. So we have carbonic anhydrase converting carbonic acid into CO2 and H2O here. What is what goes in this box, how much CO2 is present here? What is the amount of CO2 that is present here? And I'll give you a hint. It's not in units of millimolar or milliequivalents. It's in units of millimeters of mercury. 40, right? Because 40 is a normal, typical serum CO2 partial pressure. Plasma CO2 is 40 millimeters of mercury. And since CO2 can go wherever it wants, this partial pressure of CO2 is 40 millimeters of mercury. So let's see, so what happens to this CO2 when it is formed by this carbonic anhydrase? Where does it go? It doesn't stay in the urine. It goes wherever it wants, because that's what CO2 does. And so this CO2 and this CO2 are the same CO2. Just because this fluid is moving here, remember, this CO2 could be up here, and then it diffuses into the cell and becomes this CO2, right? Just because this is below here, this is true throughout this fluid, and so CO2 can diffuse into the cell. So this CO2 is part of this CO2. It's not the only part of that CO2 is the CO2 that diffused from the blood, but this CO2 and this CO2 are they're the same CO2. Okay, so this CO2, carbonic anhydrase, works in the opposite direction and makes carbonic acid, which dissociates into bicarbonate and protons. Now, the book makes it look like there is one transport mechanism for protons to move through here. But you know of one of them. I'm going to give you the other one. So, what are the mechanisms by which protons can be pumped into this urinary fluid? The sodium proton exchanger that I told you about, and a proton ATPase. So there is a proton pump that hydrolyzes ATP and pumps protons out, and there is the sodium proton exchanger that is part of this. It's part of the reabsorption story for sodium, and it's part of the excretion story for protons. Okay, so put in this box here the sodium proton exchanger and the proton ATPase. Those are the two mechanisms that are embedded in this one cartoon arrow. Okay, so similarly, there are two mechanisms that are embedded in this cartoon arrow for getting bicarbonate across the basolateral membrane. You know one of them. What is that? The sodium bicarbonate co-transporter is one of these. And there's another one that you do know because it is highly expressed in red blood cells also. The chloride bicarbonate exchanger. So in this box, there is a sodium bicarbonate co-transporter and a chloride bicarbonate exchanger. Here's the way it should look. So let's follow bicarbonate. Bicarbonate was filtered, and then it got converted into CO2, the CO2 diffused, it got converted, and the bicarbonate got reabsorbed. So that's the pathway for reabsorption of bicarbonate. Filtration, conversion, reverse conversion, and then transport into the interstitial space, the peritubular space, where it is picked up by the capillary, peritubular capillaries. So that's the complicated flow for reabsorption of bicarbonate. So let's work with this. Here it is, our friend the Davenport diagram. We're trying to unload how renal compensation for respiratory acidosis or renal compensation for respiratory alkalosis can happen. Okay, let's do this as a set of clickers. Okay, let's see. Yeah, well, okay, set of clickers, we'll get there. Let's do this one first. Which of the following enzymes or transporters is an important part of how the renal tubules control blood pH? You know we're focusing on bicarbonate. There are really two ways to correctly answer this question. You could choose them all, or you could not choose B, because, I mean, B is partly correct because the sodium potassium ATPase is what sets up the sodium gradient that features into this transporter. So that's why B could be circled, but it is not directly participating. So that's why I put it as a dotted line. If you circled them all, you're not wrong, and if you didn't, if you crossed out B, you're not wrong. Okay, that was pretty easy. Let's do another one. Altitude sickness, which is hyperventilation, is treated with a carbonic anhydrase inhibitor called diamox. Not always, sometimes. What's the mechanism that would make this drug effective? Why is this drug effective? So let's go back to the Davenport diagram. Hyperventilation is respiratory what? Alkalosis. Hyperventilation is moving from, blowing off more CO2 than is necessary, going from PCO2 of 40 to PCO2 of 20 millimeters of mercury. So this is hyperventilation. That's what happens in altitude sickness. Now, why would a drug that inhibits carbonic anhydrase be helpful? Do this as a clicker. I don't know why it goes from its to theys. It doesn't, you know, there's more than one carbonic anhydrase. So first two are referring to diamox, the next two are referring to all of them. It's the same. That's not a distinction here. That's a glitch. Okay. Show me your answers, please. This is hard. This is getting hard. I appreciate this. So the best answer is A and B. let's go back to the Davenport diagram. Here's hyperventilation. The renal compensation for hyperventilation is to try to lower plasma bicarbonate, moving along this curve, lowering plasma bicarbonate, increasing excretion of bicarbonate, right? If we are lowering plasma bicarbonate, we're excreting more bicarbonate. So going in this direction is more bicarbonate excretion. The kidneys do this by themselves. That's one of the responses to hyperventilation. And the drug accelerates, and helps, gives another mechanism for that. Now let's go back to the cartoon here. Why would inhibiting carbonic anhydrase help us to excrete bicarbonate? Well, right here, so if we don't have this carbonic anhydrase, then more of this bicarbonate stays bicarbonate, and so more of it gets excreted, because it is not converted to CO2. And remember, we depended on it being converted to CO2 to get from here to here. So by converting it less, more of it stays in the urine, more of it is excreted. Similarly, by inhibiting this carbonic anhydrase, whatever CO2 did get there from the plasma, say, less of it is converted. There is less protons being transported. So less of this can happen, and less of this can happen. So we have less reuptake and less reabsorption. Those are both the same thing. Less reabsorption means more excretion. More excretion is what we want. Drugs help us to excrete more bicarbonate. So they increase the excretion of bicarbonate, and by definition, anything that increases the excretion of bicarbonate is going to increase the clearance of bicarbonate. Those go hand in hand. That's just understanding what clearance is part of the answering of this question. They make the urine more acidic? No. Do we want to make it more acidic? We want to make the plasma more acidic. We don't want to make the urine more acidic. We want to make the urine more alkaline, so that the plasma can become more acidic. And they don't affect ventilation. Is that helpful? I mean, that was a hard question, but I am going to ask you, I'm going to ask you on the problem set, and I might ask you on the exam to work through these kinds of perturbations, whether the perturbation is a genetic anomaly or a drug effect that's perturbing the natural system and having an effect on biology. And in this case, we're talking about bicarbonate and blood pH. Tynan?
Can you remind me what ameliorating is?
Oh, ameliorate means making better.
Okay.
The word ameliorate means to make it better. Exacerbate means to make it worse. All right, let's do another one. Which of the following would be a helpful response in metabolic compensation during hypoventilation? So now we're talking about asthma or some other disease that causes hypoventilation. Which of these would be a helpful and ameliorating biological response to hypoventilation? And I'm giving you the Davenport diagram to think about it. Most of you are getting this right. So A and C are the correct answers. Somebody walk us through. So you got it right, you know how to do this. Somebody explain to the class how you got to those two true statements and the two true. Well, let's do it in two pieces. Somebody explain why A is a true statement.
Looking at the Davenport diagram,
Okay.
if you have hypoventilation, which is respiratory acidosis, you compensate
Respiratory acidosis.
by increasing the plasma bicarbonate.
Yep.
So in order to get to the bicarbonate, you need to have carbonic anhydrase create more carbonic acid, which would then turn into bicarbonate.
Right. So we want more carbonic anhydrase here in order to make more CO2 here. So more CO2 is here, more carbonic anhydrase here is making more bicarbonate here for more pumping of more recovery of bicarbonate. Wait, yeah, increased expression. We want more recovery of bicarbonate, more reabsorption, less excretion. Okay, somebody explains why C is true. You all got it right, so you know why. Just put it in words. Addie again? Addie?
Yeah, sure. So the sodium bicarbonate co-transporter is on the basolateral membrane, right?
It's
And
part
so
of this thing here, right?
Yeah, it's that one over there. And so if you have an increased expression of that, then more of the bicarbonate will be reabsorbed into the plasma.
Exactly. So both of those are biological responses. This is not a drug response. This is a biological response to hypoventilation. Your kidneys change their expression. And the mechanisms by which the kidney detects oxygen is complicated. That was a Nobel Prize thing this year, the oxygen sensing of cells. So how they detect hypoventilation is complicated, but they do, and then they respond by increasing the expression, transcription and translation, of the genes for carbonic anhydrase and the sodium bicarbonate cotransporter. So would somebody say why? Well, I guess B and D are, if you know A and C are true, then by definition B and D are false. Okay, so this is the way to be thinking about this. And the reason I like this stuff is it's combining the kidney with the lungs, which you may not have known that there was anything, that there was any interaction between those two systems until now. Well, and you heard about it in the respiratory section, and now you are understanding the molecular and pharmacological mechanisms for that relationship. Okay, let's do another one. Which of the following would be helpful in the response to a helpful response in metabolic compensation during hypoventilation? It's the same question, they're just different answers. So which of these would be helpful? This one's a tricky one. I think. I hope I got it right. Let's see. I think it's A and D, not A and C. Okay, let's walk through this. Increased so it's the same question. We're trying to recover, reabsorb more bicarbonate as a response. We've got respiratory acidosis, hypoventilation. We're trying to reabsorb more bicarbonate in order to increase plasma bicarbonate. That's what we're trying to do. We're trying to do this. we can increase the expression of the chloride bicarbonate exchanger, the same mechanism that Abby talked about, except now we're talking about the chloride bicarbonate exchanger instead of the sodium bicarbonate exchanger. Decreased expression of the sodium potassium ATPase could potentially raise remember the sodium potassium ATPase is pumping sodium from here to here, from cytosol to outside. If we decrease expression of the sodium potassium ATPase, we tend to make cytoplasmic sodium higher. More sodium bicarbonate co-transport, more excretion of the sodium, of the bicarbonate. So that one was a trick. Do the cells actually do that? I have a feeling they don't. I think the expression of the sodium potassium ATPase is constitutive, it is more or less maxed out, and I don't think the cells actually decrease the expression of it because it would have lots of negative side effects. So I don't think that happens, but it could. In principle, it's a thing that could happen, which would be a helpful response. It would be helpful. I'm not sure it actually happens. All right, let's take a break, and we're going to switch to potassium. Potassium. Reabsorbing of potassium is important. Excretion of potassium is important. Sometimes we need to excrete potassium, and there's going to be a clicker that we'll get to that. So potassium is complicated because sometimes we need to reabsorb all of it like we do with sodium all the time. And sometimes we need to excrete potassium. And the reason why we sometimes need to excrete potassium is that elevated blood potassium can be fatal. It can cause fatal arrhythmia. For example, there was a kid, this is now 20 years ago I heard this story. There was a child who had sort of an unnatural appetite for salt. It was sort of pathological intake of salt. And so his parents, being concerned about excessive salt intake, used what is called light salt, which is it's a 50-50 mix of sodium chloride and potassium chloride. And so he started ingesting huge amounts of potassium chloride, and his serum potassium went up to the point where he died, heart arrhythmia. So the point is, another point is, when the state injects a lethal combination of drugs for, what are we calling that? Capital punishment. The mixture of drugs that is used for capital punishment is a sedative to put the person to sleep. I forget the second one. And the third one is potassium chloride to cause a fatal arrhythmia. Does anyone know what the second one was? Sure. It's a sedative. Oh, I think it's a muscle paralytic to prevent respiration. So a sedative, an overdose of a sedative will also cause hypoventilation. A muscle relaxant to cause hypoventilation, to stop breathing. And potassium chloride, this is the point of the story here. Potassium chloride to cause excessive serum potassium, which will cause the heart to stop. So the point is, even though you need to sometimes excrete potassium in order to keep the serum potassium at the appropriately low level. Remember, you've got a lot of potassium inside your cells, but the serum potassium is relatively low for millimole person. So we have mechanisms for reabsorbing it when we need to, and we have mechanisms for secreting it so that we can excrete it when we need to. So let's take this apart. Let's not worry about the percentages. The points are in blue. You can reabsorb it in the proximal tubule, in the collecting tubule, and in the distal tubule, actually. You can secrete potassium when necessary by the conditions. And so what is excreted can range from a small fraction of the filtered load, that's what load here is, filtered load, a small fraction of the filtered load, to more than what was actually filtered, because it's actively, not only is it filtered, but it's actively secreted by the distal tubule and by the collecting tubule. Okay, so let's talk about those mechanisms here. So we've got another one of these guys. And there's a lot of information here, but we're going to simplify it so that it makes sense. Okay, so remember, here we are in the proximal tubule, here's the ascending limb of the loop of Henle, here's the collecting tubule, and here's the collecting tubule. So it's slightly different from the sodium images, but the same idea. Lumen, interstitial or peritubular space, apical membrane, basolateral. Okay, we're oriented? All of them have the sodium-potassium ATPase. Right? They're still there. It's the same cells. We're still talking about the same cells that we talked about for sodium. So we have the sodium-potassium ATPase in all of them. The two things to know about how potassium is reabsorbed in the proximal tubule. Remember, we have that possibility of reabsorbing up to 80% of the filtered potassium in the proximal tubule. The two things to know is that there's a potassium chloride co-transporter in the basolateral membrane, and potassium can get dragged along with the water. Remember, this is a leaky epithelium. Water is flowing paracellularly, and potassium can get dragged along with that water. Everything else on this slide you can cross out. So we don't have to worry about that, although this is part of this. We don't have to worry about this K channel. We don't have to worry about these charges. I mean, you can leave this K channel here, that's fine. But the point is, potassium is transported out in the basolateral membrane. And it makes this sort of futile cycle here. The sodium-potassium ATPase is pumping it in, and then it's leaking out. It's kind of wasteful. This is part of why the kidney is a major consumer of your ATP. Okay, so that's what's happening in the proximal tubule for the reabsorption of potassium. In the ascending limb, this is the same story as the sodium story, except that remember, from the sodium story we talked about the sodium-potassium 2-chloride co-transporter. That was part of the sodium story, but it's also part of the potassium story. Potassium is pumped into the cell in combination with the sodium that is going down its concentration gradient. So potassium is pumped into the cell, and then it leaks out through this potassium channel. The rest of these things you don't have to worry about. So potassium is being reabsorbed here by co-transport and then leaking out. Okay, so that's reabsorption of potassium. I mean, it's not as hairy. This book makes it super hairy, but if you just look at what, some of it, it makes some sense. Okay, so now we need to talk about how the collecting tubule can excrete or secrete potassium. There is well, actually, let's skip this one. Just cross out this whole thing because it's confusing. Let's just go straight to this principal cell of the collecting tubule. Okay? Remember, we have this ENaC sodium channel that is co-expressed, it's expressed sort of in tandem with this potassium channel. And this, I said this was like an exchanger, but it's not an exchanger. But it acts like an exchanger. For every sodium that is pulled in this way, there is a potassium that leaks out that way. And so this potassium leaking out that way would contribute to potassium excretion. And it would be, you know, here's a way. Potassium is pumped into the cell and then leaks out. There's the path for potassium excretion. Pumping from the blood, you know, from the peritubular fluid into the cell, and then leaking out through this potassium channel in exchange for the leaking in of the sodium. In addition, so I do want you to know this guy, and in addition there is a potassium chloride co-transporter that is another pathway for potassium to be transported out of the apical membrane. And so potassium is pumped in by the pump and then transported out by the potassium chloride co-transporter. So in the collecting tubule, when necessary, we increase the expression of this and this. We increase the expression of this transporter and this channel in order to increase the secretion of potassium when necessary. If we're in a low potassium situation, those two transporters, this channel, well, mostly it's this transporter, is the expression of it is downregulated. And so we have less potassium excretion in the collecting tube. And then all we have to worry about is, you know, the mechanisms for reabsorption that we talked about. So that's really all you need to know for potassium. I know it's not nothing. And so your challenge is to put these stories into your brain. And my recommendation for how to put these stories into your brain is to narrate slides like this in your head, explaining what you need to explain, whether you're explaining sodium reabsorption, or potassium reabsorption, or potassium excretion, or bicarbonate reabsorption, or bicarbonate excretion. Think of what you want to explain. Maybe draw a blank one of these and put in the elements of the story. I think the way to learn this stuff is to narrate the story and maybe even, you know, write it as an outline so that it gets into the writing part of your brain and is not just the listening part of your brain. This is an example of the sort of thing that is really hard to pack into your brain, and active learning, writing it out, talking it out, telling the story to your friends, even if they don't want to hear it. That's part of the way to learn it. And just by reviewing, just by looking at it and saying, oh yeah, I know all that. It's easy to deceive yourself that you know it when in fact you don't. That's my recommendation. And I do know it's a lot of information, but you know that I'm interested in you knowing molecular mechanisms, and that's what we're doing here. Okay, let's do some more clickers. Let's do the first one. The best answer is A. Sodium potassium, most of you put up A, but some of you put up some other ones. Sodium potassium ATPase, the pump, is expressed in the basolateral membrane all over the kidney. So that's important in all segments. Sodium glucose cotransporter is only expressed in the proximal tubule, so B is false. Sodium potassium 2 chloride cotransporter is only expressed in the ascending loop of Henle. So that's false. Paracellular water permeability is only really true in the proximal tubule. So that's false too. Okay, here's now. Why did humans evolve molecular mechanisms for only reabsorption of sodium, but for reabsorption or secretion of potassium? Why did we do that? I think the best answer to this one is seal. So you may not believe it, but we evolved in an environment where it was easy to get potassium from our food and very difficult to get enough sodium from our food. This is not the world we live in, but this is the world we evolved in. We evolved in a world before there were salt mines, before salt was everywhere. I mean, and where we were eating meat, and we were eating fruits, and we were eating seeds, and we were eating grains, which are all rich in potassium, and not rich in sodium. Sodium, salt, was precious. What's the root of the word salary? Saline, salt. There, in the Middle Age, even as late as the year 1000, 1200, people were paid in salt. It was that precious. You had to, I mean, you need a certain amount of salt in your diet, and it was hard to get until we learned how to mine it, and then it was not hard to get. And now we live in a world where we have excessive sodium chloride, but that's not the world we evolved in. So we evolved in a world where potassium was rich. We were eating a lot of potassium, so sometimes we had to excrete it for the reasons that I talked about before. But where sodium was almost always precious, and we needed to conserve it as much as possible. So let's go through them. Sodium is more important to cells than potassium? No, that's just not, I mean, they're both equally important. Sodium is more abundant? No, we said that that's false. Sodium tastes better than potassium? Yes, that's true, but that's not why we evolved that. Have any of you ever tasted potassium chloride? It's bitter. It's salty-ish, but it's bitter. It doesn't, I mean, it's not enjoyable to taste potassium chloride. But we get a lot of potassium from fruits, nuts, grains, and meat. Okay, let's do another one. So this is, now I'm starting to transition into understanding about water, because your problem set, which is due on Sunday, remember, is also going to be asking you about diuretic drugs. And you can look up on the internet what a diuretic drug is supposed to do, but the bottom line is a diuretic drug is to help patients excrete water. So now we're going to talk a little bit about water. Mannitol is an osmotic diuretic. This means it increases urine flow, diuresis, via an osmotic mechanism. Here's a little bit more information. Mannitol is freely filtered at the glomerulus, but it is not reabsorbed by any segment of the renal tubules. Which of the following is a true statement? many of you are getting it all right. They're all true statements. Mannitol, remember that creatinine was a good measure of GFR because it was freely filtered and not reabsorbed, which is true of mannitol as well. So mannitol is freely filtered and not reabsorbed, so it could be used to measure GFR. It's not very much, but it could be. Mannitol is an osmotic, this is really the key point. Mannitol is an osmotic diuretic because it's a solute. Any solute that appears in urine is going to be accompanied by water. So if mannitol is a solute in urine, if you put mannitol in urine by giving mannitol to a person, it's freely filtered, so it's going to get into the urine. It's going to be a solute in urine, and that's going to tend to make the urine volume more because water is going to follow solute. Maybe there's the statement I want you to walk away with here. Water follows solute. And so if you put a solute in the urine by filtration, water is going to go with it. And that's what makes it a diuretic. And it's an osmotic mechanism because it's not being pumped, nothing is being pumped anywhere. Mannitol was getting there by filtration, and water was getting there by osmosis. Mannitol is not a substrate for the sodium glucose cotransporter. That's got to be true because if it were a substrate for the sodium glucose cotransporter, it would be reabsorbed by the proximal tubule. You could say, well, maybe not. If it were, you could poke holes in this one because you could say, well, what if it were a substrate for the sodium glucose cotransporter, but it weren't a substrate for the glucose uniporter? Then it would not get reabsorbed. That would be poking a hole in the point here, but that would show me that you understood it. Did you see how what I was talking about? You would have to get mannitol across both the apical membrane and the basolateral membrane in order for it to be reabsorbed. It's not reabsorbed, so it means it is not a substrate for the sodium glucose cotransporter, and it is not a substrate for the this glucose uniporter. Mannitol does not follow a paracellular reabsorption path. It's true. If it did, it would be reabsorbed by the proximal tubule because that's where paracellular transport and water dragging things along like potassium can happen. Okay, so that's the kind of thing that I want you to be thinking about, especially for the problems that do on Sunday. Water follows solute. Okay, here's another one about diuretics. Most diuretic drugs that increase urine production, increase urinary excretion of water, are used to increase sodium excretion. Which of the following is a true statement? I think A, B, and C are all true statements. I think most of you got that right. Diuretic drugs don't affect GFR. That's not a target for diuresis. And if anything, they would increase GFR, not decrease it, in order to get more water in the urinary fluid. We got another one. Okay, let's do another one. We got another 5 minutes. Which of the following drugs would be effective for increasing sodium diuresis, sodium excretion, sodium clearance? all would be effective. So inhibiting the sodium glucose cotransporter would inhibit sodium reabsorption in the proximal tubule, more sodium, less sodium reabsorption, more sodium passing out in the urine, that's sodium diuresis. Same story for all of these things. Inhibiting the transporters that reabsorb sodium. A would be a bad diuretic. Yeah, A would be a bad diuretic because that we would now excrete, not only excrete sodium, but we would excrete glucose, which is something we don't want to do. So if you end up in the pharmaceutical industry and your boss tells you, we're gonna start working on a drug to, a diuretic that inhibits the sodium glucose cotransporter, somebody should say, wait a second, that doesn't seem like a very good drug target. We're not, that's not gonna be an effective drug because of the side effects. Right from the start, you know that would be a bad drug target. Okay, I think we're done. All right, so remember the problem set. I'll see you in lab this afternoon.